So welcome everyone to our Science Twilight lecture. I'm Hayley Smith and I'm the Faculty Outreach and Events Officer, and I will be your host today. So any problems with the sound or the session or any questions or comments you've got, please put them in the chat to the panelists, and then I'll make sure I help you throughout the session so that you can enjoy it and take part. We are recording this session, but none of your comments or chat or names in the participant list will be in the recording. It would just be the panelists, the presenters and the presentation screen that will be used in the recording. And then we can share this with you after the event if you'd like to see it again. So just before we start, I just want to go through a bit of housekeeping with you. So anything you put in the Q&A or the chat box can be seen by all panelists. So please do not share any personal information. And as I mentioned, if you've got a message on the chat box, please do it to all panelists or to everyone in the session so that we can make sure we pick it up and it gets answered. I'd now like to introduce Lauren, who's going to talk to you today and give you an introduction to cancer biology. I'll just do a screenshot with you. So welcome, Lauren.
Hello, thank you for the introduction. Welcome everybody, and thank you for attending. I'm just going to get my slides on so we can start. And here we are. I hope everybody can see the slides.
Yes, we can see it, Lauren.
Okay, great. Okay, so today I'd like to tell you a little bit about the science behind cancer. And this will be all about the molecules that are involved. I'd like to tell you a little bit about myself first. I'm very passionate about understanding how cancer occurs. How does a normal cell become a cancer cell? And I've been teaching a course at the University of Greenwich for many years. At one point, the textbook that I was using to teach my course was written by a person that was about to retire. And I panicked and I said, what am I going to use for my class if I don't have a textbook? And so I thought of the idea of writing a textbook myself. And it is my students who actually gave me the idea of how to write the book. So I used to teach cancer biology, like most people, giving you the basics throughout the year. And then the last lecture of the course, I used to talk about therapeutics. How was this information applied to new therapies? And I noticed that all the students got very excited and enthusiastic when talking about the therapies. So I decided to write a book where every chapter was linked with the basic biology to the therapies. And that idea was taken by Oxford University Press, and I had to start writing. The project took a turn, which I didn't expect, because I've just finished writing the 5th edition. So I have been writing for quite a number of years. Another project that I got started on was because of feedback on Amazon for the textbook. And the feedback was from actually a patient who had cancer. And they were trying to understand the molecular biology, but you can tell that they didn't have a background. And so this very much inspired me to write a book for a general audience called Why Millions Survive Cancer. Okay, so that's my background. And I'd like to show this slide now to show basically faces of cancer. And I hope that you see that there are familiar faces on this slide. Most people know Kylie Minogue, who had breast cancer, Lance Armstrong, who had testicular cancer, and Steve Jobs, connected with Apple, unfortunately died from pancreatic cancer. But this slide reminds us of several things. One is that things are changing because first, we're talking about cancer. Secondly, people are living with cancer. So Kylie is wonderfully carrying on her career. Lance Armstrong has a couple of different types of careers. We won't go into that now. And again, this number of 3 I'd like you to take note of. And that is cancer affects 1 in 3 people over their lifetime. So I'd like you to think about perhaps your friends or family, okay? Those numbers are severe, Okay, so this is why cancer touches everybody. So I'm going to pose the question, what is cancer? If you had to write the definition for a textbook, what would you write? Perhaps you can add a few lines in the chat, just to think about what is cancer. While that's coming through, I'll draw your attention to this photograph here, which is showing you two cancer cells, which are rounded up against a background of normal cells, and the normal cells here are stained in blue. And so you can see a physical difference of cells in culture, in a tissue culture dish, because of their shape. Cancer cells round up, and normal cells are more triangular, and they are attached to the bottom of the dish. Okay. Do we have anything interesting in our chat, Haley?
So we've got uncontrollable cell division, and then we've got uncontrolled cell division as a result of DNA mutation, and then we've got the abnormal growth of cells in an uncontrolled manner.
Thank you very much. Those are excellent contributions to the definition. So thank you, audience. That's part of the definition. So that's our first part. Cancer is a set of diseases characterized by abnormal cell growth, but it's not the full definition. So if I ask you, what's the difference between cancer and a benign tumor is not cancer, or if you ever had a wart on your foot, if you are a witch, you may have a wart on your nose. That is also abnormal cell growth. So what's the difference? What's the difference between abnormal cell growth and cancer? Anything else we need to add something to this definition? Any ideas in the chat?
We'll give them a chance to type. So okay. we've got one.
Oh.
So cancer is spread from the region it originated.
Fantastic. And that completes our definition. So very much that is the answer. Cancer is a set of diseases characterized by abnormal cell growth and the ability to spread from a primary site to other sites in the body. And we call that process metastasis. Okay, so here's another question for you. What is cancer a disease of? We say Alzheimer's is a disease of the brain, and anemia is a disease of blood, osteoporosis is a disease of bones. Cancer is a disease of, anybody want to put their answers in the chat?
So we've got cells. Let's see if we get any others. Another one for cells.
Great. Okay, so cells is a good answer, but not the best answer. It is cellular, you're right, but cancer is actually a disease of your genome, okay? It's the genes that give rise to the abnormal cell growth. So as you know, genes carry the chemical instructions that code for proteins. And proteins serve as cell parts, and they carry out numerous functions. They can code for enzymes, for example. And genes are passed down to daughter cells every time a cell divides, okay? Now, you may remember the central dogma of molecular biology. This is such an important foundational equation that you all will become familiar with as you study biology. Does anybody know what the central dogma of molecular biology is? Use your chat, please.
I'll give them a chance to type in. We'll wait for some responses.
Thank you.
not got any at the moment. It's all, oh no, sorry, we have. So we've got DNA to RNA to proteins.
Fantastic. There's an A student. Thank you very much. So that is DNA is transcribed into RNA and is translated into protein. And again, I can't overestimate how important that is. Okay, so really what is this telling us? Well, that growth factors, things that cells need to grow, are actually proteins, and they're made from our genes. If we get mutations in our genes, in the DNA, that can lead to overactive growth factors, which is a way that you can get cancer. Okay, so genes have a very important underlying role in cancer. Now, you may know that all cells except for the egg and sperm cells contain 2 alleles of every gene. And this slide just reminds us, it's a little cartoon of the difference between healthy cells, how they grow in a very precise, regulated manner in order to balance the net number of cells in the body. Cells also, when they get older, they can die by a specific program called cell suicide or apoptosis. In cancer, what happens is there is an accumulation of mutations, and the mutations affect growth so that you end up with uncontrolled growth. Okay. Now, going back to the 2 alleles for every gene, we are reminded of Mendelian genetics. Okay, so Gregor Mendel was a monk who studied the quantitation of how traits are inherited. Okay, and he studied peas, and most people know of his experiments where if he crossed 2 round heterozygous peas, which meant that they had a dominant and recessive, then the offspring would show a recessive progeny. Okay, so here you have all your dominants with the big R, which is rounded seeds, but you also have rough, okay, the rough skinned peas here. Okay, so those experiments led us to the very important essential or important dogma that you have 2 alleles per trait, and there's something called dominant versus recessive. Okay, and the same is true when we talk about cancer genes. So there are 2 major classifications of mutated genes that contribute to cancer formation, also known as carcinogenesis, okay, the making of cancer. Those types of genes are called oncogenes and tumor suppressor genes. And then they have another term for genes before they're mutated. They call them proto-oncogenes. So these are normal genes that can be mutated to become oncogenic. Okay, now generally oncogenes are dominant and tumor suppressors are recessive. So this diagram just illustrates that concept. Here you have an oncogene, and if you have a mutation, that the expression of that gene becomes more, or it gives rise to a more active protein, that is dominant, and you get growth without regulation, so that leads to abnormal growth. With a tumor suppressor, you need 2 mutations. These tumor suppressors are usually proteins that have a role in policing the net cell number in the body. So they basically inhibit growth most of the time, and that's why they're named as a tumor suppressor gene, okay? And if you have 2 mutations that knock out this function, then you're going to get, again, abnormal growth, okay? And sometimes you can have a tumor suppressor where their role is, let's say, in something like DNA repair, where half the quantity will also lead to abnormal cell growth. So we call that haplo-insufficient, haplo meaning half. Half the amount gives rise to abnormal growth in some tumor suppressors, okay? So the main thing from this slide is that oncogenes act in a dominant manner, and most tumor suppressors act in a recessive manner. Okay, you need two mutations. Okay, so people often ask if cancer is inherited, and the answer is it's only very rarely inherited. Okay, you can inherit an increased risk of cancer, but most cancer results from an accumulation of mutations over your lifetime. And that's why cancer is a disease of getting older. And you can see in this graph, on this side, we have the percent of people with invasive cancers. And on this axis, we have age group. So you can see the cases are fairly low when you're young, and they increase quite a lot as you get older, over 60. Okay. And again, that's because the accumulation of mutations over time can give rise to cancer. Okay, so some chances for audience input, please. Can you name several agents that cause cancer? And the challenge is to see if we can come up with all of the agents that I have on the slide before I show you. Okay, so see if you can just list all of the agents that cause cancer that you know of. The one in the picture is probably the most obvious. Okay, smoking.
Oh, so we're busy now. So we've got smoking and UV light, high energy radiation, alcohol, some viruses, eating high cholesterol foods, obesity, asbestos. See if we get any more. Air pollution, radiation. I think I said that. Did I say that one? Radiation.
That's great. Well done. Okay, we'll clarify some of those. Thank you, Haley. Thanks very much for feeding back. Just one question before I show you what's on the slide. If we were to be in a bubble and we could block out smoking, pollution, UV, could we still get cancer? What do you think?
We've got 3 yeses so far. 4 yeses.
Great. Thank you. And that's a good answer. Okay, so let me reveal what's on the slide. And you've done very well. There we go. So UV, you've mentioned X-rays, you said radiation, so that's fine. Cigarette smoke, did we say alcohol? Great. Alcohol, obesity, I know. Chemicals, and that's from things, everything from pollution to industrial chemicals, chemicals even in food. Infectious agents, I know you mentioned, so there's some bacteria and some viruses that can cause cancer. And the last point was from the question, I said, can you get cancer if you're in a bubble? And that is, the answer is yes, because our body makes reactive oxygen species just from breathing. We make these products, which can damage DNA and lead to mutations. Okay, so those are the agents that cause cancer. We will talk about diet a little bit later. Specifically, there are metabolites of cholesterol. I know somebody mentioned cholesterol in the chat. That can be carcinogenic. So again, that can be related to sort of chemicals. But we will talk about diet. Okay, so agents that cause cancer are called carcinogens. Okay, how do they work? Well, basically, carcinogens damage your genes. And it's quite a simple process. Many bind to and mask the chemical instructions of genes. So that means your A, T, Gs, and Cs, right, of the DNA sequence. And that masking causes the replication machinery to make a mistake. They can't see the real letter that's behind. And as a result, they put in the wrong letter, and that leads to permanent changes called mutations. Okay, so if these mutations are genes involved in cell growth, specialization, also called differentiation, or cell suicide, they can cause cancer. Okay, because it's these processes, cell growth, differentiation, or cell suicide, that affect the net cell number in the body. Okay, so I'll have a quick question. If you have a mutation in the gene for hemoglobin, will that give rise to cancer?
So we've got one yes and one no at the moment.
Okay, thank you. I think we'll stop and use that. We have both possible answers. So the answer is actually no, because hemoglobin has nothing to do with cell growth or specialization or cell suicide. Okay, hemoglobin's function is to carry oxygen in the blood, so it doesn't affect net cell numbers. Okay, so mutation in hemoglobin may give you another disease, but it won't cause cancer. Okay, this is one of my favorite cartoons to show how cigarette smoke causes cancer. Okay, note there are 81 different chemicals in cigarette smoke that are carcinogenic, that cause cancer. Okay, 81 different chemicals. What happens is the smoke is breathed in. Of course, it's your lung cells that are exposed mostly to the smoke. And those chemicals basically stick to your DNA. They mask the code. So right now, we don't know really what's underneath that letter. So when this DNA goes to replicate itself, when the cell goes to divide, during replication, they'll make a mistake. And that mistake will lead to a mutation, a change in the code. And that can change the protein, again, going back to our central dogma of molecular biology, right? A change in DNA can lead to an abnormal protein. Okay, and that can then lead to cancer. Okay, so basically the principle is quite simple of how chemicals cause cancer. And again, the more mutations you have, the more you smoke, right? The more likely you're going to hit a gene that's involved in growth, differentiation, or death. Okay, let's talk about cases in the UK. As you can see here on this side, you have females. So the top three is breast cancer, colon cancer, and lung cancer. And in the males, the top are prostate, lung, and colon cancer. Okay. It's interesting, there is a website that you can use, and you can get this information for any country in the entire world. And we do this in my class, my cancer biology class, and students choose any country they want. And it's very interesting to see the changes. So the top three, for instance, if you look in Kenya, lung cancer is not in the top three. It's not even in the top five. The reason? They don't smoke. They don't smoke cigarettes. Okay, so there's a lot about lifestyle that comes into this type of epidemiological data. So we know lifestyle matters. Okay, a very important point. Some cancer deaths can be prevented. And this is a very, very good example. The smoking bans were a brilliant thing and probably saved more lives than any drugs we now have approved. This started in 1990 in a small little town in California called San Luis Obispo. And there the mayor made the first public smoking ban. And that ban traveled across the United States, eastward, it crossed the Atlantic, it reached the UK, it crossed Europe, and further beyond. Okay, and now China also has bans on smoking. Okay, is there any evidence that the stopping of smoking has helped decrease cancer? Well, I think this graph is very obvious. And you can see that, again, on the y-axis, we have the deaths per 100,000 males, and here we have the years. So remember I said in 1990, we started the smoking bans. And you can see what has happened to the death rate in males in lung cancer. Very sharp drop. Okay, now that is very drastic. That means that all of these numbers of people who died because of lung cancer, right, could be prevented just from not smoking. And the slide also shows you some other interesting trends with other types of cancer. But of course, this is very, very strong evidence that not smoking saves lives. Okay, we can see some other cancers have decreased. We have some cancers actually a little bit on the rise, such as pancreatic cancer, liver cancer, and these are probably due to the rise in obesity. So we have seen we have several remarkable defense systems against damage caused by carcinogens. So we have DNA repair. If our DNA is damaged, we have molecular pathways that can fix that damage. The thing is not to over bombard the system. So if the more damage you give, they're more likely that the DNA repair may miss one of these mistakes, okay? We also have, as a defense, cell suicide. And I'll talk about one of those that you're probably very familiar with in a moment. We have detoxifying enzymes. You all heard about detox, right? Detoxifying enzymes is helping you to get rid of poisons, in this case, carcinogens, antioxidants, and of course, the immune system. This slide just shows us again how UV radiation causes skin cancer, right? So again, it's your skin that is exposed to the sun, and that causes a special type of mutation known as pyrimidine dimers. Pyrimidines are your T's and C's in your DNA. So if they're two T's together or two C's together, the sun will link them in an abnormal way, which distorts the helix. And as a result, again, when the cell goes to divide, it will make a mistake and put in the wrong letter. Now, I think many people have the experience that when you're in the sun for the first time of the year and you get a sunburn, and then a few days later, you can actually see your skin start to peel, okay? That is actually our defense against our damage that we did with the UV. So that peeling of the skin is apoptosis, or cell suicide, okay? Your body has decided that those cells are damaged, and so much damage we probably can't repair it all. So instead, the body triggers cell apoptosis, which kills all those damaged cells. Better to kill the cells than to get cancer. And we all know the importance of using sunscreens because we have UV radiation, which is absorbed by pigments in the sunscreens. So we can make lifestyle changes that can reduce our risk of cancer. And now the next slides are actually showing you what's recommended by the World Cancer Research Fund and the American Institute for Cancer Research, okay? They recommend to be lean, okay? Because as we know, obesity is on the list of carcinogenic agents. And also to be physically active. And these two, right, both need to be followed. You can't be a skinny couch potato, okay? Because physical activity also causes certain changes in the body to help defend ourselves against carcinogens. So be lean, be physically active, eat 5 portions of fruit and vegetables a day. I think that campaign in the UK is brilliant. I think everybody is aware that eating fruits and vegetables helps prevent cancer. Avoid sugary drinks and energy-dense foods, again, because that's helping to prevent people from getting overweight and obese. Limit your intake of alcohol. Alcohol is a known carcinogen. Limit the intake of red meat and avoid processed meat. Limit salt and avoid moldy grains and legumes. Okay. It is known that the mold that can grow on peanuts gives rise to one of the most toxic carcinogens called aflatoxin. And we're very lucky in the UK, in the USA, they screen for this mold in all our peanut butter products. But there are other parts of the world where they don't screen. So that can be a big source of carcinogens. The advice is also to meet your nutritional needs through the diet. Okay, don't rely on the perfect pill, the perfect vitamin. Okay, and the other advice is that mothers should breastfeed because, again, this is something that guards against breast cancer. Okay, so as I mentioned, we all know about eating fruits and vegetables as something that can prevent cancer. And we know that they're antioxidants, but more importantly and more recently, we found out that components of food can turn on and off genes that protect us from carcinogens. Basically, they turn on your detox genes. Okay, that's so important that I emphasize it that way. This is a little bit molecular down here, but the take-home message is the components in green tea, components in broccoli, actually can go into the cell and turn on genes that code for your detoxifying enzymes, your detox enzymes. Again, these enzymes help you to excrete chemicals more readily from the body, again, so you have less exposure. So they're protecting us. So as you can see here, there are many different fields of science that can lead to a better understanding of cancer and contribute to developing new treatments. Okay, so just from our talk today, you can see how bioscientists are involved, biomedical science, nutrition, pharmaceutical science, if we talk about making new drugs, sports science, important, chemistry, and also environmental science. Now, of course, prevention is better than a cure. And there are some strategies out there that we can use to prevent cancer. Of course, not smoking is one of them. We mentioned that bacteria and also viruses can be causative agents. And of course, we have antibiotics as a means of getting rid of bacteria. So in certain regions where carcinogenic bacteria are endemic, they have done trials to try to eliminate the bacteria to prevent cancer. We also know things like aspirin are very important and guard against inflammation. And inflammation is a causative agent of cancer. And so there are some agents there that are being used in people who have a predisposition or a tendency to get some of these cancers from an hereditary condition. And we know that some viruses cause cancer. I'm sure you're all aware of the human papillomavirus, hepatitis B viruses. Okay, human papillomavirus causes cervical cancer. Hepatitis B causes liver cancer. And we have vaccines against both. And of course, both are being used. So again, we have vaccines that can prevent cancer. That's excellent. Now, to talk about the final part of this talk, I'd like to make you aware of a new area that's showing so much promise in the field called immunotherapy. And now immunotherapies are drugs that do not target the tumor, but instead they boost your immune system for your immune system to fight the cancer. Okay, so this is a different type of strategy that we've used in the past, and it's showing to be very, very powerful. What we have been using for a while now are therapeutic antibodies. And antibodies are part of your immune system, but we've been using them as a tool, as a therapy. So we can design antibodies in the lab, and basically they have great specificity for individual molecules. And so we can make antibodies that target sort of the bad proteins from the mutated genes on cells, and they act as magic bullets. They actually can recruit immune cells to help kill these cells that they're recognizing, and they can also help get rid of these sort of misformed proteins, okay? And one example of this type of therapeutic antibody is called Herceptin. And Herceptin is approved for breast cancer, and it's been used to treat more than 1.3 million patients worldwide since 1998, okay? And there are several other therapeutic antibodies that we use to treat cancer. Now, what's exciting right now, and as I'm sure we're all very grateful for the COVID vaccines, a lot of that science has been building for quite a long time, and people are also using information to make cancer vaccines. And again, remember that cancer vaccine is getting your own body to fight the bad agent, in this case, the tumor. And they're trying to make cancer vaccines that are therapeutic, which means it will treat a cancer, or you can make a vaccine to prevent a cancer. And as we just discussed, the human papillomavirus vaccine will prevent cervical cancer. Okay, so that's a preventative vaccine. A vaccine is made up of an antigen, which is what you want your immune cells to recognize, and something called an adjuvant, which basically is a general stimulant of the immune system. Okay, so here in your vaccine, you inject antigen and adjuvant. The antigen is picked up by some of your immune cells and processed so that they introduce that antigen to the real fighters of your immune system, and those are the T cells, okay? Because the T cells then can go through the body, and when they see that antigen again, such as on a tumor, they can kill the tumor cells, okay? And that is the rationale for making vaccines. And again, the same rationale is for the COVID vaccines right now, right? We're injecting either RNA to make viral protein or DNA to make viral protein, parts of the protein. We're not injecting virus, we're injecting part of a viral protein that we want our immune system to recognize and then be able to defend ourselves against. Okay, so those are just gonna, yeah, I think that's good. So that is the summary of vaccines, and I just have to say that the immunotherapies right now are giving very, very dramatic results for some cancers that really haven't had much hope in the past. And in 2018, the Nobel Prize was awarded to scientists who really made great contributions to the field. Okay, I hope you have enjoyed the talk. Thank you very much for listening, and I'm happy to take any questions. I also note here is my email address. I welcome any questions or comments if you'd like to write to me. Thank you very much.
Thank you, Lauren. There is a question in the chat already for you. So if I read that one to you while others are posting their other questions. Okay. So it's, why can the DNA not look at the opposite strand to see what the missing base is when it is hidden by a carcinogen?
Okay, thank you for that question. The binding of the chemical to the base is actually masking. So the whatever recognizes the base can't read it. So it just cannot. The way replication happens is that each strand is separately copied. So the strand that's not damaged will be fine, but the strand that has the mask on it will be read improperly. Thank you.
Thank you. So we'll just wait for some more to come in. So if you've got a question for Lauren, then please either use the chat or the Q&A box, and I'll make sure that they get asked for you.
I also have, if you'd like, some review questions if you want. So if we run out of questions on the chat, we can try some questions.
Let's see. I'll give them a minute just to see if anything appears, because it takes a while to type everything in these sessions.
Of course. Thank you. Thank you, Haley.
Okay, so we've got a question here for you. So how did you get into cancer research?
Thank you for the question. I just like to study biology. So I just followed my passion. I studied biology. I studied for a PhD in New York on cell and developmental biology, where I ended up doing a lot of molecular biology. I studied gene expression in New York. And then I came to London to do a postdoc at the Ludwig Institute for Cancer Research. So that's my area. That's where I first was exposed to the cancer field. I worked on something called newt limb regeneration. So you may know about newts in your garden, these little green critters, but they can lose their arms and regenerate their arms. They're actually all of their limbs. They can even regenerate their jaws, and they can regenerate parts of their heart. So they're amazing animals, and they're vertebrates. So we were very interested in why these vertebrates, like us, can regenerate their arms, but we can't. And so we started to look into molecular factors, which allows them to regenerate their limbs. Again, an interesting project before I came to the University of Greenwich.
Thank you, very interesting. Just wait and see if we get any more. If not, we can go back to your summary questions.
My advice for your careers is follow what you're interested in, and it will take you to roads that you probably are not aware of now. But if you're doing what you like, then that's a good life.
Okay. We have got another question here, and I apologize if I don't pronounce all this right, because science isn't my area. So we've got some cancers affect the metabolism of cells, how glucose and oxygen is handled. What potential paths for a cure might we see there? And then also, is cell approximation therapy potential for leukemia and brain cancers? That is in the chat, so I apologize if I've not pronounced that right.
No, thank you. Let me answer the first question, because it's a very good question. There are what's called hallmarks of cancer. And this was put forth by two great scientists in 2000. And in 2011, they revamped their hallmarks and they added reprogrammed metabolism. So it's a fairly new aspect of cancer biology that people are very interested in. And you're right that there are changes due that cancer cells actually take up glucose a lot more than normal cells. And it's the basis of what we know as of PET scans, positron electron tomography. And that is actually seeing cancer cells taking up sugar more than normal cells. So yes, you're right. They are looking at metabolic differences between normal cells and cancer cells and trying to inhibit cancer-specific enzymes versus normal cell enzymes. So that's a great question. They are working on it. I'm trying to think if there's anything. There are definitely candidates in clinical trials now in that area. And your second question, sorry, Haley, I hate to ask you to read it again. I forgot.
That's okay. So it was, is cell approximation therapy potential for leukemia and brain cancers?
I'm not sure. I'm not sure what that second word is. Brain. I'm just.
I can spell it to you because
Sure.
I'm not pronouncing it right. So it's ASPHY Xiation.
Okay, thank you. I think you're talking about things like hypoxia, lack of oxygen. So there is a very important tie with what we call angiogenesis, which is that tumors try to stimulate blood vessel growth to supply them with more oxygen and more nutrients that they need compared to normal cells. Okay, so I hope I am answering your question. There is a great, and there are approved drugs that block this process of angiogenesis. Okay, and that the rationale is that you try to starve the tumor. So by blocking their blood supply, you create cells that don't have enough oxygen, don't have enough nutrients, and you hope that they die. Okay. One of the drugs that act in this way is called Avastin, and it's an anti-angiogenic drug. It's not specific for brain cancer. It's used for cancers where blood supply is important. Now it's being approved for several. I think colon cancer was one of the ones that it was first approved for. Brain cancer is tricky because of the blood-brain barrier, and some drugs may have difficulty crossing them. And again, people are working on how to get some drugs across the blood-brain barrier more readily. Okay, so again, those are areas of new research. But good questions. I hope I answered your question.
Thank you. So we've got some more for you. So the next question is, how likely do you think it will be that breath biopsy becomes mainstream in cancer detection?
Oh, that's a very good question. The thing is to identify compounds that can be distinguished at a high enough level between cancer cells and normal cells. I don't know of any that are close to being approved, although I know people are working on that aspect, and it would be an absolute wonderful thing if they can get it to work. But again, quantity, they have to get to a good sensitivity. Anything that's non-invasive would be wonderful to prevent people having to go through a biopsy. There are a lot of diagnostic tests coming through, screening tests, perhaps looking at pieces of DNA in the bloodstream. That is invasive, but it's not as uncomfortable as a biopsy. So I think there's a lot that can be seen in just a quick blood test. So that is a big improvement as well. There are tests coming out of testing products in the urine. So they have one of the most specific tests out now for prostate cancer, where they're looking for what's called a non-coding RNA in urine. And again, that is a big improvement from prostate biopsy. So yeah, good question to think about different ways that people can be diagnosed for an earlier diagnosis, as we know, is the best thing for having the best outcome. So yeah, great question. Thank you.
Thank you. So just to let you know, we've got 10 minutes left, and I've got another question here for you. So it's a follow-up question from one before. So it's follow-up on the sustained angiogenesis hallmark of cancer. So how do the cells persuade the body to grow those blood vessels?
It's a very good question. So there's something called the angiogenic switch. So think of a light switch on and off. And we have factors in the body that both inhibit and stimulate the growth of new blood vessels. The growth of new blood vessels is very important as an embryo, but in the adult, we hardly use that process except for two cases. One is if you have a wound, then you want the growth of new blood vessels, and the other is in the case of the female reproductive cycle. So most of the time, our switch is off, but the tumors can create the positive factors. So they can turn the switch on. And some of these factors are just specific growth factors. So for example, there is a specific growth factor for blood vessel cells, and it's called VEGF. It stands for vascular endothelial growth factor, okay? And it's just a growth factor for blood vessels. And cancer cells can produce this, and they get the blood vessels to grow towards them, bringing them oxygen and nutrients, okay? Remember, the cancer cells are dividing rapidly. They undergo mutations more, and so they are trying to survive. That's their goal, trying to change the body so that they can survive within the host. They are our own cells that have gone wrong, basically, right? So cancer is not from a foreign agent. It's our own cells that have been mutated and behave abnormally.
Thank you.
Thank you, Haley.
So we have got another question that's coming as well. So it's, why do you think some of the top cancers in the UK are around reproductive organs, for example, breast and prostate?
That's a very, very good question. And in fact, it's something that I learned from doing this exercise with my class, where we looked at all countries in the world, and breast cancer is in the top 3 for most of the places around the world. Very interesting. Now, if you think about the risk factors for breast cancer, one of the things is estrogen. So anything that allows an increased exposure to estrogen, but again, estrogen is an endogenous substance, isn't it? So it seems like too much exposure of estrogen is one of the factors. But then there are things that add to that. So alcohol, actually, its metabolism is linked to estrogen synthesis. Also, obesity is linked because fat tissue can make estrogen. And it's very interesting. I think in women, in your younger years, your ovaries are your organs that make estrogen. In your later years, it's your fat cells that make estrogen. So again, interesting links between obesity, estrogen exposure, and breast cancer. I don't know if I have some convincing evidence for prostate cancer, that again, but linked to some endogenous hormones. So yeah, there's some curiosities there, but again, it's an some endogenous carcinogens.
Brilliant, thank you. We're getting lots of questions. There's another one that's just come in. I apologise. So it's that you mentioned another hallmark of cancer, the ability to cells to invade apoptosis by activating gene pathways. What are these gene pathways, and how do they render these cells immortal after generations of growth?
Okay, I see 2 areas in that question, so I'll see if I can answer them in the right way. So apoptosis, so a hallmark of cancer is avoiding apoptosis, so avoiding cell death, right? Cell death helps us to get rid of the damaged cells, as you remember in that slide about sunburn, okay? So again, there are molecular pathways that our body has to trigger apoptosis, and the cancer cells can block that. Now, one of the most famous tumor suppressor genes that we have is called P53, okay? And P53 triggers DNA repair. Remember, it's a tumor suppressor, it's gonna protect us. So it triggers DNA repair, it inhibits cell growth, and it stimulates apoptosis. If we have a mutation in P53, then apoptosis can be blocked. So there are many different molecular ways where, again, we tilt the system and we can stop something that's supposed to happen from happening, and therefore you get more cells. So if you come and join my class in cancer biology. We'll be talking about more specific molecules that I'm sure you will enjoy. Your other question was apoptosis, and what was the other one? I'm sorry, I forgot already.
So it's, what are these gene paths and how do they render these cells immortal after generations of growth?
Okay, thank you. The second part reminds me of telomerase and telomeres. And I don't know if you know about telomeres, but they're the ends of your chromosomes. And every time a cell divides, they get shorter and shorter and shorter. And so they have the hypothesis that the telomere length is linked to aging, cellular aging. Interesting aside, I'm sure you all heard about Dolly the sheep, which was the first animal that was cloned, and she was cloned from an adult cell. So all of her body cells were basically coming from an adult cell already. And there was a lot of talk about whether her telomeres were shorter than the normal sheep. And again, it's connected to cellular aging. And that does connect to replication. So when you have cells that have a shortened telomere, they'll divide a number of times and then they'll stop dividing. And that's what we see in cell culture. Okay, cells in culture don't divide forever. They'll divide, let's say, 10 times and then they'll die out. However, there are some cells that have an enzyme called telomerase, which helps to extend these ends of the chromosomes and basically keep that replicative potential longer. And sure enough, stem cells in the body and also cancer cells have high amounts of this enzyme, telomerase. So it seems like this enzyme is helping to keep that replicative potential going. So very good question. And if you're interested, have a read about telomeres. It's a very fascinating area. And of course, that's giving us a target as a drug too. People are trying to see how they can inhibit this enzyme, telomerase, in cancer cells for that reason. Okay, so great question.
Brilliant, thank you. So we are on the last 2 minutes of the session. So if we do a magic screenshot, swap, sorry.
Thank you, my audience, and thank you for your very, very interesting questions. Again, I wish you well and take care.
So then just before we all log off, hopefully, so just want to, that's all right. No, you're fine. Thank you, Lauren. So just for the audience, this is a feedback survey. I've just put the link in the chat for you as well. So we do value your feedback and it does help us improve our sessions and make sure that we're targeting them to what you want to hear. And also any suggestions for anything you'd like to hear about in the future is also welcome as well, because it just means we can put more sessions and run events with you as well. So the link is in the chat, or you can scan the QR code and access it. It will only take you a few minutes and it will really help us with our outreach provision. And then just to end the session is just so contact details. If you have any more questions or need to contact us, I put Lauren's email in the address in the chat for you as well. So obviously if you want to ask her more about the session and the topic area, then obviously she's offered and said you can contact her. We've got more events coming up as well. So if you want to come and see us at other things and find more out about the faculty or the university, then please visit us, and they're all listed on the events page, which is in the chat and also on the screen there as well. So just want to end by saying thank you very much, Lauren. It's been very interesting. And thank you to the audience as well. And thank you very much for engaging as well and putting lots of comments and answers and questions in the chat as well. So thank you, everybody.
Thank you.